Good morning, everyone, and everyone joining. It is my pleasure today to introduce our Grand Round speaker, Dr. Erin Giles, who is an associate professor at the University of Michigan. Erin started her career getting her PhD at McMaster University, where she studied insulin-like growth factor binding protein 3 and TGF-beta in breast cancer metastasis to the bone. She then went on to do her postdoctoral training at the University of Colorado, Anschutz Medical Campus, kind of my old stomping grounds. And so we have some connections there that we were just talking about. And this is where she really developed her interest in obesity and breast cancer. She then started her position as an assistant professor at Texas A&M, and then recently, a couple of years ago, made a small hop up north to the University of Michigan School of Kinesiology, where she is now an tenured associate professor. Erin's work is on studying obesity on metabolic health in preclinical models, really with an emphasis on postmenopausal breast cancer. She's very interested in how menopausal weight gain and adipose inflammation that's associated with this contribute to breast cancer risk and progression, and also on how diet, exercise, and pharmacological interventions can be used to decrease obesity-associated comorbidities after menopause. She is NCI funded to study the role of visceral fat in breast cancer risk, and has published quite extensively in this area. And I want to give her a lot of credit. She'll talk about her preclinical models today. One of the biggest challenges in sort of the preclinical rodent models of breast cancer is that we are really good at modeling triple-negative breast cancer in mice. But it is really, really hard to model, for example, hormone responsive and dependent cancers in mouse models. And really, rat models are much better at modeling this. And I believe she will talk about her rat models today. And this is sort of an area where, I always think, oh, we should study ER-positive breast cancer. And then people say, oh, you really need to use rat models for that. And then I'm like, nope, don't need to study it. So I give her a lot of credit. She is one of the few investigators using these models and doing really nice work with them, and really important work. So thank you for joining us this morning, Erin, and take it away.
Yeah, thank you. I should have you do my introductions every time. That made me sound so wonderful. So thank you for the invitation. I'm excited to give a talk and share a little bit about what we've been working on. So as Kaylee mentioned, we do a lot of work in rats. And so I thought I'd start this talk by just giving a little bit of overview for those of you who don't study breast cancer on the link between obesity and breast cancer, sort of the role of menopause in that relationship. Move on to highlighting sort of some key aspects of the model that we use quite a bit in the rat. And then I'm going to give four little vignettes here. Hopefully I have time for all four. If not, we can sort of cut through one of those, highlighting some recent studies. Most of these are unpublished. The Preventing Weight Gain After Menopause is published. This menopause timing paper should be on bioRxiv by the end of the week, I hope. This DOV study is under review right now, and then some data that we're working on in exercise and where we're heading. So for those of you who don't think about obesity quite as much as I do, I thought I'd just share the most recent statistics for the US. And I think there's, in 2022, there was 19 states where the obesity rates are greater than 35%, and an additional 3 states where this is more than 40%. I always like to highlight that this data comes from self-reported data where people give their body weight and their height. And as you can expect, most people underestimate their weight and overestimate their height. So this probably underrepresents the state of obesity in the country. And just to highlight, where we've come from, this is the same data from 2011. So if I sort of toggle back and forth, it's definitely a big color shift there. And you can see our rates are definitely on the rise. So why do we care about obesity? I think many of you probably know that obesity is a risk factor for cancer. There's 13 different cancers that were at least strongly associated with obesity in the last WCRF and AICR report. And I'll just highlight here where it says breast cancer, and you'll notice in sort of that small font underneath, it specifically highlights postmenopausal breast cancer. And so in addition to increasing the risk for postmenopausal breast cancer, we know that women with obesity also have an increased risk of metastases from these cancers. and due to that sort of increased mortality related to these cancers. As Kaylee mentioned, these are mostly those hormone-sensitive tumors, ER/PR positive tumors. There's probably a really complicated relationship if we start looking at other tumor subtypes. So for today, I'm going to focus mostly on these hormone-dependent postmenopausal breast cancers. And so we also know, in addition to obesity being a risk factor, gaining extra weight through adulthood would further increase a woman's risk, as does having type 2 diabetes. Gestational diabetes further increases the risk. So there's a lot of risk factors going on here, but what we think we can take away from this epidemiological data is that menopause is likely a window of time in somebody's life where you might be able to intervene to decrease obesity and maybe decrease their breast cancer risk. And this will become sort of important as I get to the end of the talk. I think from the recent statistics, we would suggest that about a quarter of women who are in this sort of 45 to 60-year-old age group, postmenopausal, are at high enough risk for breast cancer due to all of these factors that they could consider therapies to reduce their breast cancer risk or for breast cancer prevention. And I'll talk a little bit about a newer ongoing study we have looking at breast cancer prevention agents. So talking about this rat model that we use quite a bit in the lab, this model was developed in collaboration with Pepper Shadeen and Paul McLean at the University of Colorado. So I was a postdoc that came in on the sort of beginning parts of this project. Pepper had a lot of the breast cancer expertise. Paul has done a lot of work in the obesity metabolism space, and we decided to sort of combine models of obesity and cancer together. And our question was, could we come up with a rat model where obesity primarily promotes tumor development only after menopause, or a model of menopause? So here we start with female Wistar rats. Wistars are an outbred strain. This means that they're genetically slightly different and they respond differently to a high-fat diet. So we put them all on the same high-fat, 45% fat diet. We individually house them to limit their physical activity. We can measure food intake, do body weights, all of those sorts of things. At 7 weeks of age, so this is where the mammary gland is developing in a rat, we inject a single dose of carcinogen, methyl nitrosourea, and this increases DNA damage, and primarily in the mammary gland because it's undergoing a lot of proliferation due to the puberty. And so this induces primarily mammary tumors in these models. We do get a few off-target tumors, but primarily mammary tumors that are ER positive. So I said we place all the animals on the same high-fat diet, and here I'm just showing you a histogram of the response that we get in percent body fat. And so you can see that this ranges sort of from 8% on the left up to about 30% body fat. Again, these are animals of the same age that have been on the same diet for 18 or so weeks of age. So what we do is we select the animals that are in the lower third of percent body fat. These will develop into lean animals. We select those in the top third of percent body fat, obesity-prone animals, which develop to become obese. We then use a surgical model of ovariectomy. So we surgically remove the ovaries to model menopause. Like in humans, this increases weight gain. So in the rats, you get about a 3-week period of weight gain rapidly. And then we can follow these animals and their tumors for either short or long-term studies. And so we've published quite a bit on this model, and so I put some of those references below. And what I thought I would do is just highlight a few takeaways from our early studies. And so, as I said, we were trying to ask, can we get a model where we only get tumor promotion after menopause? And here we're showing data from this 2010 paper where we started with a similar number of tumors at the time of ovariectomy. And if we ovexed the lean animals compared to the obese, followed them for, I think, 12 to 14 weeks afterwards, what we were able to see is that in the obese animals, you had fewer tumors that regress, more that progress. And because it's carcinogen-induced tumors, you can get these spontaneous tumors developing. We got an increased number of new tumors that developed in the post-ovex period. So we think this does a pretty good job of modeling what we're seeing in the human. And the data that I don't have time to show you today was that one of the strongest predictors of tumor progression in the model was the rate of weight gain during this 3-week period of rapid weight gain after ovariectomy. So the follow-up study that we did for this was to look at that 3-week window and say, what is it about this menopausal weight gain that could be driving tumor growth? And so to do this, we took that same model, and during that 3-week period of weight gain, we put the rats in an indirect calorimeter. So this is a closed chamber that measures oxygen consumption and CO2 production. And we were also able to feed them a radioactive fat. So we put carbon-14 labeled dietary fat into the diet on the last day of the study. So now you can see where in the body the dietary fat is going. And then we also gave the animals a single injection of 2-deoxyglucose that was labeled with a second radio-labeled tracer, tritium. And so now we can see where's glucose and where's fat going in the body and how might this be driving tumor growth. And so for anybody who does these types of studies, the one thing you'll note is that if we're looking at animals in an indirect calorimeter during a period of rapid weight gain, that weight gain can also drive or dictate where nutrients are going. So sort of to compensate for this, what we did was we stratified the animals into those that were in either low or high energy excess during the last 24 hours in the calorimeter. And so as you can see here, we have a nice sort of 2x2 design where we have lean animals that were either overfeeding about 10 kcals extra per day versus those who had high energy excess, about 25 extra kcals per day. So you can think of this as sort of a mild overfeeding versus like Thanksgiving dinner. Then same thing in the obese. And what this allows us to do now is separate, are the effects that we are seeing due to either the underlying phenotype, either being lean or obese, or those extra nutrients that they're consuming during this period of weight gain. And so what we found is that if we look at the normal mammary gland and we look at the lean animals on the left here, what we see is that if they're undergoing high energy excess or consuming lots of calories, there's a significant increase in the amount of glucose that's being taken up into the normal mammary tissue. So this is exactly what you would expect, sort of insulin-stimulated glucose uptake. And if you look at the obese animals, you can see that the amount of energy excess does not drive glucose uptake into the normal gland. However, if we look at the nearby tumor in the same mammary tissue, what you can see is that the lean animals have no response to this energy excess in their tumor glucose uptake. Whereas if we look at the tumors from the obese animals, it's those under high energy excess that have increased glucose uptake. So this suggests that if the mammary gland is not responding normally, and or if there's differences in the tumor, we can drive glucose uptake into these tumors. And what we've been able to show is that this is associated with increased proliferation, tumor number, and a lot of changes in these metabolic pathways in these tumors. So that was sort of the working initial studies that we did with these models and gave us this working hypothesis that to increase breast cancer development and progression, you need both this underlying obesity and the metabolic impairments that go along with that, as well as this positive energy imbalance or extra nutrients that are induced by this overfeeding during the period of postmenopausal or post-OVX induced weight gain. And so if this is in fact true, this would suggest that we could either come in and improve the underlying metabolic impairments or prevent that energy excess, and these should be able to decrease tumor growth. And so we tested this directly by using both metformin, which is a most commonly used anti-diabetic drug that would improve metabolic control. And we also did a study where we looked at preventing weight gain during this postmenopausal period by adjusting the diet. And so this is sort of the study design for that. We did a few other interventions in the study. So similar study design, what you've seen before. At the time of ovariectomy, animals were randomized to either ad libitum feeding, so as much as they wanted of that same high-fat diet, metformin in the drinking water, weight maintenance, where we gave them a bolus of food every day to keep them at their pre-OVX body weight. And we had another question that was related to inflammation in the mammary adipose tissue, and to sort of address that, we treated the animals with ibuprofen. And so if you were sort of paying attention at the beginning and heard that Kaylee say that I was here at the Michigan School of Kinesiology, what you might understand is I also have some interest in exercise. And we think that exercise could sort of improve all of these. And I'll get to that sort of at the end where I think these studies could be going. And so these studies were not designed for a head-to-head comparison of all of the groups. We didn't have the study numbers for that. But I thought it was useful to put all of these sort of tumor outcomes on the same graph so you can see them relative to each other. And so this is a change in tumor volume from the time of ovariectomy to 8 weeks post-OVX. And like we often see in this model, when we surgically remove the ovaries, we get rid of estrogen pretty quickly. The tumors start to shrink a little bit. And then in the control animals, they always tend to sort of bounce back. And if we kept this going out for several more weeks, you would see a significant increase in tumor growth. As you can see in sort of this orangey red color, the weight maintenance interventions significantly decreased tumor growth. The metformin was pretty good as well, and the ibuprofen was somewhere in the middle. And when we looked at the number of new tumors that developed over the post-OVX period, you see this similar sort of dose response where weight maintenance had the best effects. Metformin, while it did decrease tumor numbers, was not quite as good as the weight maintenance intervention, and the ibuprofen, again, is somewhere in the middle. So the metformin study's been published, and so I'm not going to talk a lot about that today. I'll follow it up with a second question we had about metformin, but I thought I'd highlight some of the findings from the weight maintenance paper. This was in collaboration with Elizabeth Wellberg, who's now at Oklahoma with her own lab, and then talk a little bit about this window of opportunity with metformin, which is work that my student Karen Corletto was involved with when she was a graduate student in my lab at Texas A&M. So if we look at the weight maintenance, as I said, we gave animals a bolus of food every day to keep them at their pre-OVX body weight. So here I'm just showing you the body weights over the post-OVX period. The lean animals in all these graphs are in blue, the obese are in red, and you can see that here, both the obese and the lean gain a significant amount of weight as they go after this post-ovariectomy period. So OVX induces weight gain. And in the open squares for both groups, you can see that we did a pretty good job keeping them at their pre-OVX body weight. I think all animals were within 3% of their initial body weight. And this was sort of an unexpected study design, but that it worked out nicely that the lean ad libitum animals ended up at the same body weight at the end of the study as the obese weight maintained. So that was sort of a happy accident. As we look at women who go through menopause, we see that not only do they gain weight and gain adipose tissue, but they also change where this adipose tissue is deposited. And so you often think of somebody going from a pear shape where they have a lot of their adipose in their hips and thighs to sort of an increase in visceral fat, taking on that apple shape with the abdominal adiposity. And so we can look at this sort of indirectly in the rodents as well. When we looked at visceral fat, this is a combination of the mesenteric around the GI tract, the retroperitoneal, which goes back up around the kidneys, and then the gonadal fat. And what we see is that both the weight-maintained lean and weight-maintained obese had a reduction in their visceral fat, in addition to overall a reduction in body weight and total adiposity. We also looked at sort of systemic markers of metabolic health. So here I'm showing you some livers. This is adipophilin staining, so marking lipid droplets in the livers. And you can see that both the lean and the obese have a significant amount of lipid in their livers. This is not unexpected, given that they're all on a high-fat diet for their entire lives. And you can see that the weight maintenance intervention significantly decreases the brown staining here. And this is quantified in the graph below. We also saw that we improved insulin resistance in these animals. So here I'm showing you HOMA-IR, a homeostatic model of insulin resistance. This is a ratio sort of glucose and insulin in the animal. And we can see that there's a significant reduction in the insulin resistance. We also see a significant reduction in leptin. This is an adipokine secreted from your adipose tissue. So this is not unexpected, but it's a good finding. And we also see a reduction in circulating cholesterol as well. Finally, we looked at the adipose tissue, and here I'm showing you sort of a marker of adipose inflammation. So this is CD68 staining by IHC for macrophages, and they're forming these crown-like structures. So these macrophages can come in and surround sort of unhealthy or dying adipocytes. And we can see that, well, we had no effect on the number of crown-like structures in the lean. This is probably because they had a somewhat low level to start with. But in the obese animals, we have an increased level of crown-like structures in the controls, and this is significantly reduced with a weight maintenance intervention. We think some of this is driven or correlated with adipocyte size. So here I'm showing you the range of the diameters of adipocytes in the lean animals on the top and in the obese on the bottom. And what you can hopefully see quite easily here is that in the control animals, we have fewer small adipocytes and more big adipocytes compared to the weight maintenance, where you have an increase in the number of these small cells and a reduction in this sort of moderate to larger adipocytes. And so we know that smaller adipocytes are more insulin sensitive. They can take up nutrients more easily into their environment, which suggests that this might correlate with sort of those early findings where we saw that there was a change in nutrient uptake into the adipose tissue, which could be driving tumor growth. We also looked at some more systemic markers of inflammation. This was sort of a study that was done in the midst of COVID, and it turned out to be sort of a happy accident that we weren't able to do some of this in our lab. And so we farmed out some of these cytokines and got probably more data than we normally would. So here I'm showing you, this was about 100 different cytokines that were measured. We took the differences between the control and weight maintenance group, put the significantly altered cytokines into enricher to look at sort of which pathways might be involved. And you can see a significant reduction in a number of both adipokine and pro-inflammatory and pro-proliferative pathways in these circulating factors. I'm just going to highlight a few examples on the right here to show you that we have a significant reduction in TNF alpha, for example. And I also wanted to highlight FGF2 because this will sort of come into play later in the presentation where we've seen similar types of markers changing in some of our clinical samples. So overall, this weight maintenance intervention is improving both systemic inflammatory and adipokines. So when we looked at the mammary adipose tissue itself and the tumor, we saw similar changes in FGF signaling. So that was FGF2 that I showed you in the circulation. If we go back, we sort of had some data sitting in the archives where we previously looked at the expression of FGF1 in that early study where we did those nutrient tracers with animals under low or high energy excess groups. And what we happened to see for a sort of separate project was that the combination of high energy excess in the obese animals, we had an increase in the expression of FGF1 in the mammary adipose tissue. And so when we came back to this weight maintenance study, we said, okay, let's look at those same tissues. So we took the mammary glands, we stained them for FGF1, and we saw that the weight maintenance intervention was able to reduce FGF1 in the mammary adipose. We then looked in the nearby tumors, and we similarly stained for phospho-FGFR1. This is the receptor, then phosphorylation shows activation of this. And so we saw that the weight maintenance animals had less phospho-FGFR1 staining, suggesting lower activation of the FGF pathway in their tumors. And so together, this data suggests that FGF1 might be a driver of this obesity-associated breast cancer growth, and that preventing weight gain during this post-menopausal or post-OVX period could interfere with the FGF-mediated tumor growth and be at least one mechanism by which we might be seeing this beneficial effect. So sort of switching gears, I showed you this data, we asked about, okay, what happens with the weight maintenance? And the other side of the coin was, well, is it this underlying metabolic changes that happen with obesity that could be also driving tumor growth? And I said that metformin decreased tumor growth and decreased the number of new tumors. And well, metformin is a very commonly used drug, and we know that it's safe and well tolerated. It's sort of not a drug that you might want to be putting people on as a preventative strategy for very long periods of time. So the next question that we had was, could we actually target metformin to that early 3-week window of postmenopausal weight gain that we previously shown had been sort of a driver of tumor growth? And would we get the same beneficial effects as we do when we give it sort of a long period of time in these animals? And so this is a study, as I mentioned, that one of my graduate students was highly involved in. And here we don't have lean and obese animals, but what we did is we used that same model, sort of took all comers, ovariectomized the animals that once they had a sort of small tumor, and randomized them either to a control group, so this is ad libitum feeding on that same high-fat diet, or metformin in the drinking water for 8 weeks. So those are the 2 groups that you've already seen. And then we added an additional 2 groups where we said, well, what happens if we put them on metformin for the first 4 weeks of the study, sort of over that early postmenopausal weight gain period, and then take them off the metformin for the last 4 weeks. And so as I mentioned, the metformin here is in the drinking water, so this is just an easy water bottle replacement. And in the sort of control, we added an extra group where we had water only for the first 4 weeks, and then we moved them on to metformin for the last 4 weeks of the study. So we're calling these the early metformin and the late metformin groups. And so our hypothesis here was that metformin would be most effective when it was targeted to this sort of 3-week window of time, or at least as equally effective as it was with the 8-week group. Excuse me. So here I'm showing you the Kaplan-Meier curve of tumor progression in this model. And so as you can see, I hope pretty easily here, the metformin early group was equally effective as the metformin 8-week group in preventing tumor progression. Whereas those animals that were randomized to metformin late in the study had a tumor progression that was similar to the level of the controls. And similarly, if we look at the number of new tumors that developed in these animals, well, these results did not reach statistical significance. We sort of see a trend for a reduction in the number of new tumors in the metformin group. We previously published this type of data. However, a little bit surprisingly, we saw that those in the metformin early group actually had sort of an increase in the number of new tumors, whereas the metformin late had a reduction in the number of new tumors. So nothing that reached statistical significance with those new tumors, but I think it's interesting data to sort of take a look at. The other sort of unexpected finding we had here was that stopping metformin at that early 4-week period also had some maybe negative effects on weight gain and adiposity. So here I'm showing you their cumulative weight gain over the study. The metformin treated animals, when they switch, don't gain a significant amount of weight compared to the controls, but we see this sort of increase in weight gain, or at least the rate of weight gain, in those animals where we removed the metformin. Whereas if we start metformin at that 4-week time point, we can actually blunt weight gain. And we see maybe similar or even more significant results when we look at percent body fat. So here you can see that those metformin early group had a significant increase in body weight sort of at that 4-week time point, whereas starting metformin at the 4-week time point can blunt weight gain. This on its own might not be super surprising or super significant. A little bit of weight gain might not be the worst thing in the world if you were having a beneficial effect on tumor outcomes. But we also saw some sort of concerning outcomes when we looked at circulating factors. So we saw that the change in insulin from OVEX to the end of the study, we have an increase in insulin in that early metformin group, an increase in insulin resistance, and also an increase in circulating levels of IGF-1, which is also known to be tumor promotional. So together, this would suggest that targeting metformin to that early postmenopausal window could be beneficial for reducing tumor growth, but stopping it had some somewhat negative effects on their metabolic health. We do have some questions, if we phased out the timing that we were removing the metformin, did it a little bit more slowly rather than this abrupt stopping, we might have been able to ameliorate that, but that's a question for another study. So last sort of story that we have to tell here is about a cancer prevention agent that we've been doing some work with. And I think I told you at the very beginning of the talk that about a quarter of women who are have obesity or at high enough risk for breast cancer that they could consider agents for preventing that cancer. And so one of the most common agents that would be recommended for a woman in their sort of peri or postmenopausal period would be tamoxifen. And we know that tamoxifen is super effective as an anti-estrogen. It's used to treat breast cancer and is also really effective for prevention. One of the big problems and the reasons that women won't take this often for prevention purposes is that in women that are already going through menopause who may have hot flashes and other vasomotor symptoms, tamoxifen can also induce hot flashes and these symptoms. So the last thing a woman wants as they're starting to have hot flashes is go on a drug that's going to increase that risk. We also know in women who are already overweight or have obesity that there's also detrimental metabolic effects. And so these include the increased risk of type 2 diabetes and insulin resistance in women who are already overweight and at risk for this. So together, that's sort of all of the negative reasons that many times people will say, no, I don't really want to go on this drug for prevention. So one of the other options that we've been investigating, and this is in collaboration with Carol Fabian at the University of Kansas Cancer Center, is a combination of basidoxifene, which is a SERM, an estrogen receptor modulator, combined with conjugated estrogens. It's packaged or formulated as the drug called Duovy. And this drug is FDA approved already for the relief of hot flashes in postmenopausal women. And it also is used to improve bone density in that population. And some of the early preclinical and human studies suggested that there might be some anti-cancer effect of this drug. It's been shown to reduce the fibroglandular volume on mammogram And in benign breast biopsies, there's a reduction in Ki-67 and other markers of proliferation. But one of the questions we had was, what does underlying obesity do to this drug? There was very little knowledge on that. And this was particularly important given that we knew that tamoxifen had sort of worse metabolic effects in those who are already overweight or obese. And so in collaboration with Carol and her team, as well as Steve Hursting at North Carolina and Catherine Cook, who's at Wake Forest, we underwent another rodent study here looking at this drug and its response on several metabolic markers. And so here's sort of the study design. This is a little bit different than the study designs you've seen previously in that in this study, we used older ovary intact rats, trying to mimic that sort of late premenopausal or early perimenopausal period. I will tell you, we have another study that we did with ovariectomized animals. So we've done it sort of under both conditions. So here you have the same sort of study design, female Wistar rats on the high-fat diet. We gave them a carcinogen, but in this study, we did not have any palpable tumors. So these are animals that got carcinogen to increase their risk. Those that got the tumors early on went to another study, and these were the ones that had some DNA damage, but we started them on the drug before they had any overt tumors. So again, we classified them as lean or obese based on their percent body fat, and then we randomized them to either this combination of beta-doxyphene conjugated estrogens or a vehicle control. In this study, because it's a fat-soluble drug, we put it in a mixture of Nutella and gave it to the animals on a little tile bolus every day. And we followed the animals again for 8 weeks. And as I said, we did a similar study in OPEX animals. So what did we see? So here I'm showing you their percent body fat, and we see a significant reduction in both the obese in green and the lean in purple in response to this drug. We see a similar reduction in body weight, not showing you that data today, but it reduces body weight and percent body fat. When we look at fat distribution, we saw a significant but 40% reduction in visceral fat, primarily in the obese animals. Probably this is because the lean animals are not super obese to start with in this one, don't have a lot of fat to lose. When we looked at insulin resistance, this follows the same trend where the obese animals see improvements in insulin resistance. Again, this is driven by both insulin and circulating glucose in these animals. When we looked at other adipokines, so adiponectin is a beneficial adipokine secreted from the adipose tissue, known to have anti-cancer effects, whereas leptin is also secreted from the adipose tissue as sort of that negative marker, which has been known to drive tumor growth. And when we looked at the ratio, so an increase would be a beneficial change here in this study, and we see a rise in the ratio of adiponectin to leptin in both the lean and the obese groups. We then took the normal mammary tissue and we actually went back into microarrays here rather than sort of sequencing. So here I'm showing you sort of gene set enrichment data, showing you changes in proliferation markers in response to the beta-doxyphene conjugated estrogen. And you can see that whether the animals were lean or obese, we see a significant sort of about threefold reduction in some of these pathways associated with proliferation, suggesting beneficial sort of anti-cancer effects of these drugs. And then finally, we looked at some changes in the gut microbiome, and here I'm highlighting two bugs that were measured there where we see a significant increase in Odorbacter lanus. This would be a positive association to have a rise in this. It has been linked to improvements in insulin resistance and other similar metabolic effects, and a reduction in Lactococcus lacti, which similarly is a good change for this bug. And so together that suggests, I think, that this combination has beneficial effects both on whole body metabolism and proliferation at the level of the mammary gland, suggesting it might be a good drug to use for cancer prevention in this population who might not really want to take tamoxifen. And so I guess that is exactly what I just said there. The one thing that I'll add is that in collaboration with Dr. Fabian, we have a phase 2b clinical trial ongoing with this drug, where we're getting markers of insulin resistance, both at baseline and the end of a 6-month intervention, as well as breast biopsies from these women. And our preliminary data supports this similar reductions in insulin resistance, although I will say I don't think we're seeing quite the same beneficial effect on body weight and percent body fat. So it is nice that we're still seeing the metabolic effects, even if the weight loss effects are not quite as big as they might be. So in summary, we think this has some potential specifically in this high-risk population where tamoxifen may not be a good option. So finally, in the last little bit of time we have here, we're sort of switching gears, and I'm going to talk to you a little bit about exercise. So I think all of these changes in whole body metabolism, weight gain, could be ameliorated if we could include exercise. We know that most of the population doesn't exercise enough, or it's really hard to get people to exercise. But I think breast cancer patients or people who are at high risk might be a population who would really alter their lifestyle if they knew that it had some great benefits. And so there's a lot of epidemiological data that suggests that if you are a person who've exercised for most of your life, you have a reduced risk of getting breast cancer. The numbers vary based on the study you look at, but I think that ranges from, as little as 15, but as high as 60% reduction in risk. And we know that people who are lifelong exercisers have reduced mortality when they do get breast cancer. So about a 30% reduction in mortality. There's also been a lot of prospective studies in breast cancer patients or survivors of breast cancer showing that exercise is feasible. So feasibility studies have been done in high-risk women during cancer treatment and survivors. And there's a lot of data showing that exercise reduces biomarkers of risk. What I will say is we have very little data on the prospective cohorts where we've been able to look at cancer risk with exercise, just because I think the time it takes so long to do these studies, you need large numbers of patients, and people's level of exercise tends to vary over their lifespan, and it's associated with a lot of other changes. So diet and exercise go quite hand in hand, and so it's hard to untangle what is the effect of exercise versus all of those other lifestyle factors. So I think we have a lot of questions in the exercise space. Big ones are, how much exercise does somebody need to do to reduce their risk? What type of exercise is better? Is endurance better than resistance, or do you need a combination of the two? And then understanding the mechanisms. I think at the end of the day, there's a lot of people who would like an exercise pill, but also if we could understand the mechanisms, we could tailor exercise interventions to sort of reach those whatever biomarkers, milestones, trying to get the appropriate changes from exercise to reduce risk. And so work that I've been involved in is mostly sort of collaborative with my clinical collaborators. And we look a lot at cancer survivors because I think this is an easy population to study, or a little bit easier. And many of these women have obesity, and very few of them exercise more than sort of the recommended 150 minutes a week. And there's a lot of reasons why we think this may be. I think a lot of it has to do with fear, or either not knowing how to exercise, or fears of getting injured, and sort of having this unwillingness, unknowing how to go and exercise and get the best out of the exercise intervention. But what we do know is that if women can reach 200 minutes of exercise, that this can help maintain weight loss in women who are undergoing a calorie restricted diet. And we've shown that you can see a reduction in visceral fat even if there is no weight loss. We also know that in women who get at least 150 minutes a week of exercise, their decreased cancer mortality at 1 year post-diagnosis. So there's a lot of reasons to try and do this exercise. So in a study with Carol Fabian, Bruce Kimler, Steve Hursting, and their team, we looked at older sedentary women who are either high risk or were breast cancer survivors. And the question here was, if you gave women the tools, would they be able to then keep exercising on their own and see these beneficial effects? And so this was a feasibility study. They were aiming for 200 minutes a week of moderate to vigorous activity. Again, with a diet intervention, because we're trying to get a little bit of weight loss here. And the way this was done, it was that the women were given a membership to the YMCA and for the duration of the study. And they also had sessions with a personal trainer for the first 12 weeks of the study. So 40% of their sessions were with a personal trainer. The other time they were allowed to go into the YMCA by themselves, or they could just do exercise on their own. At the 12-week time point, the access to the personal trainer was stopped, but they continued to have access to the YMCA membership and had some group phone sessions sort of to keep them motivated with study intervention. Visceral fat was measured. This is one of the reasons I was super excited about getting on in this study. Visceral fat was measured, as well as a number of other cancer biomarkers. And so the question was, is this aerobic exercise intervention correlated with changes in visceral fat and other risk biomarkers? And for those of you who don't think about visceral fat that often, and just to sort of highlight, here we did an IDEXA to measure their visceral fat content. And if you have a visceral fat level higher than 1.2 kilograms, this is considered having excess visceral fat. So this is sort of a cut point. And what we found was that, sort of not surprising, but diet and exercise improve body composition. But the nice thing to see here, and I'm highlighting the visceral fat in this yellow box, is that we see a reduction in 3 weeks, which are the closed symbols for each sort of time point here. But even when the personal trainer was stopped, and we had some women who continued on until the 6-month time point, they continued to see a reduction in visceral fat, and they have very little loss of lean mass. And so we're primarily inducing fat mass here with strong reductions in visceral fat. So at baseline, there was 95% of the women had excess visceral fat, more than this 1.2 kilograms. And at the 3-month time point, this was down to 13 individuals, and they didn't gain it back in this following 3 months. We also looked at serum biomarkers of cancer risk here. And again, we're showing the 3-month and 6-month time points here. You can see, like we saw in the rodent studies with weight maintenance, we see a rise in the adiponectin to leptin ratio, a really nice rise at 6 months. And I also highlighted sort of FGF2. If you remember way back when I said, look at this graph of FGF2, we see a similar reduction in FGF2 at both time points. And again, all of these are other beneficial changes that were correlated sort of with this reduction in visceral fat. And so we think that sort of at least visceral fat could be a driver of a lot of these pro-tumorigenic cytokines, and losing weight and preventing weight gain could benefit these biomarkers and hopefully benefit tumor outcomes. So where are we going with this? Well, I sort of said that visceral fat is likely involved, but what we really want to see is visceral fat actually a driver of cancer risk? And follow-up studies of, could you use exercise to actually reduce tumor outcomes? So there's a lot of correlative data, but we haven't shown really any cause and effect yet. So sort of to highlight some data that we have from exercise studies, we thought, okay, we'll just come in with exercise and we can reduce visceral fat. And when we look at rodent models, these are male rodents, and as sort of expected, when you exercise them for 8 weeks, exercise is shown in the squares here, and sedentary animals in the triangles, and you see a nice significant reduction in body weight. Unfortunately, I think like many studies, females are a little bit more complicated than the males, and we actually don't see this significant reduction in body weight in female ovary intact animals. And I think this is because females compensate. And so we see changes in energy expenditure that sort of offset the energy expended by the exercise bout. And in this study, these were both treadmill running studies. And we see that they also increase their food intake a little bit. So it's harder for women to lose weight with exercise, and I think that is known in the clinical literature, and we're seeing this in the rodents as well. However, when we came in and did this in post-ovariectomy animals, so here we took same as our tumor model, we took lean and obese rats, we ovariectomized them, and then we randomized them to either treadmill exercise or control sedentary conditions. And what you'll see here is that this is their body weight over the post-OVX period. And while we didn't see a reduction in body weight in the lean, we see a small but significant reduction in body weight in the obese exercisers. Again, when we look at visceral fat, this follows the same trend where we see no change in visceral fat in the leans, but in the obese, we get a reduction in visceral fat deposition as well. So this would suggest that we can get these changes in the post-menopausal period, even if they don't happen necessarily in the ovary intact animals. So then our question is what does this do to tumor outcomes? We haven't gotten there yet, but what I will show you is that we've looked at measures of adipose inflammation, which we think may also be driving some of this tumor risk. And when we look in the rats, we see a reduction in CD68, which is a marker of macrophages. I showed you previously some staining for crown-like structures, which we know are pro-tumorigenic, and we see a reduction of these with exercise. We've also looked at some other macrophage markers here. So for those of you who think about macrophages, they exist along a spectrum, but they can be sort of considered pro or anti-inflammatory. And we see a reduction in both phenotypes of macrophages, at least by looking at the markers INOS and STAB1. And when this is great in a rat, but the question is, does the same thing happen in humans? And so in collaboration with Wendy Court and Kathleen Gavin, who are at the University of Colorado, we were able to get some adipose biopsies from women, both pre and post a medical intervention, medical model of menopause. So here they took women who were premenopausal. They randomized them, or they gave them all a gonadotropin releasing hormone agonist. So this is a drug that causes sort of a flare of ovarian hormones and then suppresses ovarian hormone production, puts them into a menopausal state. And we were able to get biopsies from women who were then either randomized to sedentary conditions or an exercise intervention. So similar, but a little bit different than the rat. But what we see is that exercise also reduces the number of CD68 macrophages in that adipose. In this case is abdominal adipose, subcutaneous, but a significant reduction in CD68 and a similar reduction in iNOS, so another macrophage marker. So we think that we're getting similar metabolic effects, anti-inflammatory effects of the exercise, and ongoing studies are looking at, does this actually decrease tumor growth? And so sort of a follow-up to that, Kaylee mentioned at the beginning that I've got some R01 funding from NCI to look at some of these questions. I've really had a hard time convincing study sections that exercise is the way to go, but as a sort of cause and effect question about the role of visceral fat, what we are doing in the lab is actually removing the visceral fat surgically. So here we're taking animals who are obese, with these large insulin-resistant, metabolically inflexible adipocytes. We know that when they undergo menopause, they gain weight. And by menopause here, I'm talking about the surgical model of ovariectomy. We know that visceral fat expands. This increases inflammation and insulin resistance. And our question is, if we come in and remove the visceral fat, can that reduce some of the downstream effects that are pro-tumorigenic? So what we think is happening is that there's a release of pro-inflammatory cytokines from the visceral fat. They can signal and increase inflammation in the local tumor microenvironment. We have some hypotheses here around specific subsets of macrophages, and we think that those macrophages could be causal in the transformation process of the mammary epithelial, resulting in tumor development and growth. And so the hypothesis is if we can reduce the amount of visceral fat during this period of time, we might be able to also reduce tumor growth. And so those studies are ongoing in my lab right now. We're doing those in the rat model as well as a parallel mouse model. So it's two models here combined with some in vitro experiments. And then as I've been talking a lot about weight gain and changes in body weight and visceral fat, probably some of you have been thinking about, yeah, what about all those new drugs on the market? So we also have some ongoing studies of these incretin mimetics. Specifically, we've been looking at tirzepatide in these models to see, can you induce weight loss with these drugs? And does that have the same beneficial effects, or do they have any other altering effects on tumor development? So finally, a little bit of pilot data from the lipectomy studies. So as I said, we took these animals, and at the time of ovariectomy, we randomized them either to a visceral fat lipectomy or a control group where we just took out the ovaries. So this is, the gray is all ovariectomy alone, blue is ovariectomy plus lipectomy. And what we can see here is that there's no difference in the absolute amount of body weight that is gained over the post-OVX period. However, we see a significant reduction in gonadal fat. That's kind of as expected. We surgically removed the gonadal fat, so it's a good thing that it's lower. But what we also see is that there's an increase in adipose deposition in the mammary adipose tissue here. But if we look at circulating factors, this lipectomy is able to reduce circulating glucose and reduce inflammatory markers such as IL-6. And so then our question is, well, what is it doing to the actual mammary gland? And so what we've done here is we've taken the mammary adipose tissue, we've digested it, isolated the CD45 population, so the immune cell population, because we have some specific hypotheses around these immune cells, and we did single cell RNA-seq on these. So here I'm showing you some very preliminary, hot off the press pilot data where we look at sort of the ovariectomy plus lipectomy compared to the controls. And what we can see is that the lipectomy has a reduction in this population number 0 here. Those are your monocytes. We see a reduction in the number of B cells. That's number 3 here. And we also see a small reduction in the number of NK cells. It's sort of this purple 14 population here. In comparison, we see an increase in the number of T cells with this lipectomy. So again, lipectomy of the gonadal fat, but these are effects in the mammary gland, and all the T cell populations are sort of along here. So I think the 1, 4, 8, 6, and 15, no, 18 are all different subsets of T cells that are increased. So we're starting to ask, what are those exact changes that are going on, and could those be driving tumor growth? And we have some animals that are developing tumors right now in the lab that have either had the lipectomy or just the ovex alone. So we'll have some tumor data in the coming months. That should be pretty exciting. So in summary, hopefully I've shared with you that we have a somewhat novel rat model of obesity and postmenopausal breast cancer. I didn't show you any of the staining there, but we have strong ER/PR positive tumors in this model, and we've published that. But I think there's unlimited opportunities to sort of test interventions in this model. We've only scratched the surface. What we have established is that the tumor promoting effects of obesity after menopause seems to require both the underlying obesity, that sort of impaired metabolism associated with that, as well as weight gain and excess nutrients. Visceral fat we think is critically important, although we'll have more data on that soon. And I just scratched the surface, but we have a lot of data sort of ongoing about inflammation and the role that that could play in this process. I think the perimenopausal period or menopausal period represents a time that we could target some interventions to decrease cancer risk, although there's questions about how long you need to maintain those over the perimenopausal period to actually achieve benefit. And then finally, we have these ongoing studies that are looking at the role of exercise, visceral adipose tissue, sort of glycemic control as well in there, and their effects on reducing cancer risk. And if women do get tumors or these animals get tumors, can they similarly reduce progression? So with that, I will thank all of the people who helped with this. So Karen and Ramsey and Danilo in my lab at Texas A&M did a lot of this work. And again, the models and the early work was in collaboration with Paul McLean and Pepper Shadeen and Liz Wellberg, who are all now at other places, but contributed to this significantly. And with that, I will take any questions.
Thank you, Erin. That was great. I'm inspired to
do a rat study.
So yeah, if happy to take questions. Looks like Scott has the first hand raised.
I do. It looks like my ability to turn on my video has been disabled, so you're gonna have to just look at the static picture of me while I ask my question. Oh, there we go. Squeaky wheel gets the grease. So that was a beautiful talk. I really enjoyed that. So these effects of obesity and inflammation on tumorigenesis, do you think they're occurring at the level of the actual transformation event, the epithelial cells genome being damaged and the oncogenes and tumor suppressor genes being activated in a certain way? Or do you think it's more the fitness of that transformed cell afterwards in terms of immune surveillance or ability to survive, evade apoptosis, that sort of thing? I'm trying to think of the earliest events and the exact mechanism at which this is occurring.
So I don't have a strong answer to that, but I think it could be both. And the reason I say that is we do have some data that I haven't shown you that we've done some soft agar transformation assays. And what we've done is we've taken adipose tissue from either a lean or an obese animal. We've made a lysate sort of slurry of that, put that on the cells so these are normal mammary epithelial cells, seeing if they can transform. And the adipose tissue from the obese animal increases the transformation potential. So that to me suggests at least something early on could be involved. Although I think all of the other studies that I've been showing you suggest that there's things in that once the tumor has developed, it can also decrease progression. And so I think it's a combination of the two.
So in that in vitro model that you described, there's no carcinogen being applied, it's just the adipose slurry.
Exactly. So that, they're not huge effects, but when you put in a sort of positive control, you're seeing levels that are 2/3 of what the positive control is getting with sort of the obese adipose tissue. So I think there's something in the obese adipose tissue. And my gut instinct would be that it's something inflammatory, although we haven't gotten too far with those assays. Those are the types of studies that we're actually sort of thinking about for some in vitro assays along with this visceral fat question. So hopefully we'll have some more answers to that later, but that's about as far as we've gotten so far.
Fascinating. Thank you.
Yep.
Dr. Seicha?
Hi, good morning. That's fantastic presentation. So I was wondering if you have been done or thinking or planning to do some sort of a lipidomic analysis of this adipose tissues to identify the most dominant lipid that could be potentially causing this inflammatory phenotypes.
We've thought about it. We haven't done any of it. So I think I gave a talk a few weeks ago. Now that I've been in Michigan just 2 years now, so I know that sounds like a really long time, but when you move across the country, it goes by really fast. And so we've been actually talking with some collaborators here who have the expertise to sort of do those analyses. And I think we might just try and send them a bunch of samples and see what they can help us figure out. So yeah, great question.
I muted myself apparently. I did want to ask you, the beta-doxephene studies are really exciting. Can you talk a little bit more about the phase 2 clinical trial and like the who is enrolled in that trial and what is it looking at specifically?
Yeah, so those are women that come into the high-risk breast clinic. So Carol Fabian runs a high-risk clinic. So those are people that usually, most of them have obesity, so they have a high BMI, but they've been referred to that clinic for other sort of family risk factors. I don't think any of them are BRCA mutation carriers. I'm pretty sure that they were not included in this, but I could be wrong. So they've come to this clinic for various reasons that they've been deemed high risk. And when they come there, they're getting sort of Volpara mammograms to get mammographic density. They're getting blood draws and then fine needle perialveolar biopsies, RPFNAs. So we have breast epithelial cells as well as some adipocytes from those women. And so what they're doing is they're taking all those at baseline, putting them on the drug, 6 months later getting sort of follow-up measures, and as well as sort of height, weight, BMI. I don't know that we're getting DEXAs on those, but we should have some at least biomarkers of fat distribution. And then at the end of the study, seeing how does it change. I think insulin resistance is one of the primary outcomes, as is Ki67 in the mammary epithelial cells. And so preliminary data is looking good, and I think I'm trying to remember, maybe 2/3 of the way through recruitment. The numbers are not large for that. I think Carol presented some of that data last year at San Antonio. So I think that was sort of the midpoint of the clinical trial. So it's looking good. It's well tolerated, but I don't think we're seeing the weight loss that we see in the rodents, but we're seeing beneficial effects on insulin resistance. So I think that's great. And then they're going to be sending us some of those. They've been able to take the fine needle biopsies of the breast, spin those cells down into a gel of some sort and do histology sections. And so we're actually hoping to be able to measure the size of the adipocytes. So we've done some trials on those where we can get adipocyte size from the breast biopsies as well, which is we didn't think we'd be able to do on these fine needle aspirates, but we might. So
Yeah, interesting.
yeah,
very cool.
But it seems to be the new drug. So.
Well, I'd say, these studies seem very timely given that, every time I turn around, there's an Ozempic commercial.
Yeah. There's also a team at Northwestern that has another clinical trial going on with the beta-doxephene conjugated estrogen. So,
okay. Yeah. Very cool. All right. Any further questions? If not, I think we will thank Erin for her great presentation. Lots to think about. And have a good day, everybody.